Hi, my name is Olivia Piacentini. Today is June 3rd, 2024. Through loss of yield and cost management, global pests pose a significant economic threat. Key pests such as the pink bollworm, Mediterranean fruit fly, and diamondback moths threaten to harm various crops. Conventional control methods such as synthetic insecticides and biopesticides have failed as pests have evolved to be resistant. Therefore, the field of genetics has stepped into the battle against pests through creating alternative methods for pest control. These genetic insect control methods have been supported through the use of data from mathematical modeling and laboratory and field testing. This has allowed scientists to provide insights and explore limitations. One method where genetics has improved effectiveness is in the sterile insect technique, or SIT. In this method, the release of sterile insects into target wild populations occurs. The released insects have been exposed to ionizing radiation to induce dominant lethal mutations, which results in sterility. SIT is species specific and therefore poses no risk of affecting other species in an environment. It was found to be most successful in areas where a single species is the cause of major harm. Genetics-based variants of SIT introduce a dominant lethal phenotype where homozygous males mate with wild females, causing all offspring to die prior to maturity. This results in a reduction in the population size. This allows for SIT methods to be utilized in species where high doses of radiation cause too much damage in somatic cells and tissues. Furthermore, sex specificity can enable these male-only releases through removing the repressor from the final generation, resulting in only male survivors. Another alternative method is Bt biopesticides in the form of sprays. It works as genetically modified insecticidal crops will release the Bt toxin to protect plants from susceptible pests. These transgenic Bt crops are a valuable method in managing the tendency of insects to evolve resistance. A method in which evolution of resistance can be slowed is through the high-dose refuge strategy. However, the use of high dose alone would cause rapid spread of resistance, as only homozygous insects would survive holding that resistance trait. Therefore, the refuge aspect of the strategy creates an area of non-transgenic plants that serve as hosts for the susceptible and resistant insects to mate, produce offspring that are heterozygous and killed by the toxin. However, methods are more practical, cost-effective, and sustainable in delaying the evolution of resistance when used alongside other methods. Managing resistance is an important consideration in genetic insect control for longevity of crop protection. Combining Bt plants with female lethal genetic insects is a solution proposed to delay the evolution of resistance. SIT can be used to reverse the spread of resistance in Bt crops. Offspring of release engineered males will inherit the lethal allele and a susceptible allele, which dilutes resistance. Mathematical modeling can be used to understand potential effects of genetically modified insects in an environment. It does this through revealing underlying biological processes, designing appropriate traits, predicting impact of fitness costs, informing design and evaluation of experiments, and exploring benefits. A variety of measures, such as the critical threshold or tipping point, are common models of genetic control. Modeling can also be used to determine evolution of Bt resistance over time. It revealed that with smaller refuges, resistance can spread faster. However, it is also showed that the use of Bt toxins and engineered insects produce equivalent resistance management with smaller refuge size. It is also important to evaluate regulatory, social, and ethical considerations. It is important that wider biodiversity in human health is not adversely affected by the release of genetically modified insects and plants. Through mathematical modeling, laboratory studies, and field trials, it is important to determine environmental and public risk potentials prior to the commercial implementation of these techniques. The risk of agricultural pests and disease is great, and I believe the use of genetic technologies presents opportunities for preservation of human health and greater food stability globally. Although I do think that the use of multidisciplinary testing to determine regulation, risks, and benefits, which will therefore dictate safe practices of genetic technology and pest and agricultural management is highly important in the stability of agriculture, our environment, and human safety.